This is podcast number 40. In this podcast, we're going to start talking about photosynthesis and how plants can capture light energy to make energy that animal cells are used to, ATP. And you're going to see a lot of it is really similar to the ideas that we've already talked about when we talked about breaking down glucose. In terms of where you are, we're talking about the big idea of homeostasis. This is idea of plants are providing their own food and regulating their inside environment instead of animal cells that acquire nutrients ultimately from plants in order to get their energy. This is a image of an early photosynthesis scientist that was laser etched onto a leaf. When you shine a bright light onto a leaf, it causes the chloroplasts to move away from the light. They actually avoid the light so they don't get damaged, and you get lighter areas. So this is caused by chloroplasts moving away from where the light was shined on the plant. And these are experiments done by Priestley. He used one of the earliest photosynthesis researchers about the time of the American Revolution. He was doing these experiments. And he did an experiment in a jar where it was sealed off from the atmosphere. He took some mint leaves from his garden and a candle and would light the flame using a magnifying glass so that he could not contaminate the atmosphere in here, and the plant would burn if there was a mint leaf in there. But something about the flame would change the atmosphere so that it could no longer burn. But if you left the plant in sunlight, again, without opening up this container, and then you relight the candle, it would burn again after being in sunlight. But if this was not left in sunlight, this would not happen. It would not be able to burn again. So something provided from the plant was allowing the candle to burn. We now know that it's oxygen. Oxygen started increasing in Earth's atmosphere as plants evolved. Initially, it was poisonous, but then there are mutations and adaptations that allowed oxygen to be used by cells and became adaptive. But even now, too much oxygen is toxic. In fact, large aquariums, like if you go to a public aquarium and they have this big water system, they use concentrated oxygen injected into the water to sterilize it, to kill bacteria. So too much oxygen can be bad. But oxygen is something we need for life. And we learned in a previous podcast that is because oxygen is the terminal electron acceptor in mitochondria, and eukaryotes can't live without mitochondria. Okay, so where's the oxygen that is supplied from those plants? Where's it coming from? If you shine different wavelengths of light, visible light, you all learned in some part of your schooling Roy G. Biv, so red, orange, yellow, green, blue, indigo, violet over here. If you shine different lights, wavelengths of light on bacteria that can detect oxygen, they congregate around wavelengths of light where lots of oxygen is produced, and not so much around wavelengths of light where not much oxygen is produced. Notice that is green. Not much oxygen produced in green wavelengths, and that's the plants are green because they reflect green light, not because they absorb green light. So if you look more quantitatively on how much oxygen is produced, as you expose plants to light, more light intensity leads to more oxygen production. But then if you get rid of the oxygen and you supply less light, you get less oxygen. So it's proportional to how much light is produced, or how much light is supplied by the supplied to the plant. So early experiments, like over 200 years ago, knew that plants were providing something, giving off something that candles needed to burn, and that was oxygen. And then later experiments showed that different wavelengths of light were not equally good at allowing the plant to produce oxygen, and that more light led to more oxygen production in plants. If we now look at the visible light spectrum from wavelengths of 300 nanometers to 800 nanometers, these would be different colors of light, which colors of light support photosynthesis. Okay, this is the oxygen-loving bacteria that we saw before. If we look at chloroplast, the organelle inside plants that absorbs light, they have their peak absorbance in the 480 range, and then it falls off on either extreme. But chloroplast contains different sorts of pigments. You get to see these different pigments showing up in the fall. So the greens dominate, the green pigments dominate, but there are other pigments that are in the leaf, and they have different colors, like the carotenes are oranges, the luteins are yellows, and so on. These, after the green pigments die off in the fall, you get to see some of these pigments. It's also how the pigments interact with sugars in the plant to produce some of the bright reds that you see in maple leaves, for example. But in a leaf, you have more than one pigment, and each of those individual pigments of pigment is a molecule, and each of those individual pigments has an absorption spectrum, wavelengths of light where it absorbs a lot of light, wavelengths of light where it doesn't absorb a lot of light. They tend to overlap in terms of where they absorb light, and they tend to, in general, not absorb green light, instead reflect green light. If we look at the anatomy of a chloroplast, this is something I want you to be able to draw, and when we're done in the next podcast talking about chloroplasts, I'll go over the handout I have on photosynthesis that includes a drawing of a chloroplast. But a simple drawing of chloroplasts, remember chloroplasts have three membranes, unlike mitochondria that have two outer membranes in chloroplasts, and then one inner membrane that's all folded up. Now, this book talks about the thylakoid space inside. So this is a membrane that's all folded up. It is the membrane is that black line, and then there's a space that it contains, and that's the green line inside the black line. I call that space, and other photosynthesis scientists call that space the lumen inside the thylakoid lumen. So I want you to use that term lumen, but you'll see in the figures in the book it'll say just thylakoid space. They mean the same thing, but I want you to know that term lumen. So 2 membranes on the outside, 1 membrane on the inside is all folded up, and it creates several spaces. So there's a space that is inside the 2 membranes, but outside the innermost membrane, and that's called the stroma. Okay, that's the space outside the thylakoid membrane, and then the space all the way inside 3 membranes is called the lumen. Okay, and it'll be important to know where things are happening in the chloroplast, just like it was important to know where things were happening in the mitochondrion. Okay, if you look at an electron micrograph of a chloroplast, here's a chloroplast here. Okay, it's hard to see the 2 outer membranes, but it's easy to see the inner membrane, and it can be in spaces where it's not stacked or spaces where it is stacked. The stacks are called grana, and you'll see that on the diagram in the next podcast. Double outer membranes, the stroma is the space on the outside, and where these pigments are that are going to capture the light, they are embedded in the membrane of the thylakoid. The thylakoid is the 3rd innermost membrane that's all folded up, creates a space. In that membrane, in the bilayer of that membrane, are pigments. So here's chlorophyll A, and chlorophyll B has a slightly different structure. And then the carotenoids and luteins also have a different sort of structure. This has a heme, which contains a magnesium ion. It's a very similar structure to the heme in your hemoglobin, the protein that carries oxygen in your blood. But in hemoglobin, the one in your blood, the metal that is inside that ring is iron, and in chlorophyll, it's magnesium. In the structure of thylakoids, there are many pigments, and those many pigments are called the antenna pigment complex. So there are many pigments. Each of these pigments can absorb light, and they can transfer energy from one pigment to the next. So this is it's called space filling diagram. This is called carbon backbone diagram. If we look at a simpler diagram, here's the membrane bilayer of the thylakoid, and these different pigments are embedded in this membrane, and the whole thing is called an antenna complex. So what happens is light will strike the thylakoid membranes, which has pigments in it. Those pigments absorb that light, and their electrons in their outer energy shell of their atoms get excited, and they go to a higher energy level when they get excited. Okay, so the light that shines on them might not be at exactly the right angle for any particular photo pigment, but something in this antennal complex will be able to absorb that light. You can see it's not hitting all of the molecules here, but some of them. And so it's sort of like a satellite dish in that it's collecting signals from the whole area of the dish, and then focusing that signal to a wire inside the satellite. So you're going to see that these antenna pigments pick up the energy of the sun, and then they focus that energy to a reaction center, chlorophyll. Okay, green light it's not absorbed, it's reflected. the absorbed light is sent, the energy is sent to the reaction center in the physical center of this antenna complex. So let's say some violet light is absorbed by this pigment. Their electrons get excited. They jump to a higher energy level. When they fall back down to their original energy level, that energy gets transferred to the reaction center. So all these guys are picking up the energy from light, and that energy is being transferred to the reaction center. And in the reaction center, what's going to happen is an electron is actually going to leave that reaction center and move along an electron transport chain, just like you saw in mitochondria. So if you look at chloroplasts that are exposed to light, you can see that they have a pH change when they're exposed to light. The stroma, when exposed to light, the pH increases, and in the thylakoid space, the pH decreases, and in the dark, it normalizes. So hydrogen ions are pumped from the stroma into the lumen of the thylakoid. So I like to think about this like those stacks of membranes back here. Okay, these stacks of membranes here are like stacks of pancakes, and these are called grana. Who makes the best pancakes? Grandmas do. So this is grana with an N instead of grandma with an M. But you have a stack called the grana. Inside that stack is the lumen. And if your grandma is really fancy, she makes stuffed pancakes, stuffed with, chocolate or Nutella or jam or something like that. And that's what happens during photosynthesis is protons get stuffed into the middle of that little tiny space into the lumen, and then they're going to come back out into the stroma. So I know that's a pretty tortured reference, but hopefully you're going to remember it because it's so silly. Again, they come out of the lumen, so the pH goes up in the stroma. They come out of the stroma and go into the lumen. So the pH goes down in the lumen and goes up in the stroma when exposed to light. But when the lights are off, that effect goes away. Okay? So the pH change in the dark, not exposed to light, not very much. pH change when it is exposed to light, big change. Okay? All right. So what causes the pH change? Well, we're going to start off on the left side of this diagram in what's called photosystem 2, and then we're going to move to photosystem 1. These things were named in the order in which they were historically discovered. So this one was discovered first, and then this one is discovered second. What a photosystem is its antenna complex that's going to absorb the light, and then the electron transport chain that are going to transfer the electrons. That whole thing is a photosystem. And we're going to start on this side just because it's easier to follow the path of the electrons. So photons from sunlight are absorbed by the antenna complex. The antenna complex pigments get excited. Their electrons go to a higher energy level. They come back to their original energy level, and then the energy that is released when they come back is funneled to the reaction center, in this diagram shown in the center. And when it's funneled to the reaction center, instead of electrons just getting more energized and then less energized, these electrons actually physically leave that molecule. And the electrons that physically leave that molecule go to an electron transport chain, just like we saw electrons that physically leave NADH are electrons that physically leave FADH2, go to an electron transport chain in mitochondria, but this is in the chloroplast. So these electron carriers, which are in the thylakoid membrane, they pass electrons from high energy, they got their energy from the sun, to a little bit lower, a little bit lower, a little bit lower, and so on energy. Okay? And then while this is going on, they're losing their electrons. Sunlight is also coming into a different photosystem called photosystem 1, and the same sort of thing is happening. Their antenna chlorophylls are absorbing light, they're passing that energy to the reaction center chlorophyll, and this chlorophyll kicks out an electron, and it passes down an electron transport chain. This electron is eventually going to get passed off to a molecule called NADP. Okay, it's different from NAD, which you saw in glycolysis and the citric acid cycle. This is NADP. It's a similar molecule, but not exactly the same because it's got that P on the end. And it gets reduced by the electrons coming off this chain and forming NADPH. So, the terminal electron acceptor, the last thing to accept electrons in photosynthesis, is NADP to form NADPH. In mitochondria, which is called respiration, the terminal electron acceptor is oxygen. Oxygen is the last thing to grab the electrons. In chloroplasts, in photosynthesis, the last thing to grab the electrons is NADP. All right, let's talk about this a little bit differently. So electrons are leaving this photosystem and they're falling down this chain, and then they're arriving in this photosystem. Well, while that was happening, this photosystem also had electrons leaving. So the one that left here gets filled in by the one that's coming from photosystem 1. And then this electron travels down this chain and then it ends up on NADP. Okay? So we have an electron that leaves here, travels down here, and then fills in this spot. This spot was left empty by the electron that left here. Now this is called P680. This is called P700 because the wavelength that gets this the most excited is 700. The wavelength that gets this the most excited is 680. And you're going to see in a second, just like we saw during electron transport in mitochondria, as these electrons are moving downhill in terms of energy, that's used to move protons from the stroma to the lumen. And the same thing over here. As these electrons are moving down their energy gradient, that's used to move protons from the stroma to the lumen. Okay? So this creates a hole of electrons in that reaction center. Those electrons travel downhill to fill the hole that was created by the P700 photosystem. Okay, but we still have a hole in the photosystem 2, a hole of electrons. So you can take one electron away from something if you go through this once, maybe 2 electrons away from something, but eventually you're not going to be able to take any more electrons away from that. You got to fill that hole. Okay? So this is called non-cyclic electron flow. If you talk about photosynthesis in another class, they might talk about cyclic electron flow. We're not going to talk about that in this class. Okay. So we got to fill that hole here. Okay, that from photosystem 1, that electron left. Where do you fill that hole from? You fill it from water. So plants have an enzyme and a catalyst metal that work together to split water. That water that gets split gives electrons to fill the hole. It also gives off protons that help make the thylakoid space have a lower pH. And then it gives off oxygen as a waste product. So when plants split water in order to fill the hole, they end up with oxygen as a waste product, and that's why plants give off oxygen. It's because they're doing photosynthesis. They need electrons to fill their hole in their photosystem. They get that from splitting water, and oxygen is left over. Okay? All right. So as I mentioned before, as the electrons are going down their photosystem and losing energy, that energy that is lost is used to move protons from the stroma into the lumen, both in photosystem 2 and in photosystem 1, from the stroma into the lumen. So into the middle of that pancake, stuffing that pancake. Okay. Now we have a pH gradient where the pH is lower inside the lumen than in the stroma. The hydrogen ion concentration is higher in the lumen than in the stroma. And with that pH gradient, that's energy, and we can convert that energy into ATP. Okay? So in the dark, plants are not making much ATP in their chloroplasts. Okay? I'll come back to that in a second. But if you turn on the light, they make lots of ATP. All right? And if you turn on the light, the pH in the lumen goes down. Okay? And that low pH is tied to making ATP. How do they make ATP? They do it the same way that mitochondria do. They have an ATP synthase that looks just like it does in mitochondria. Hydrogen ions go from the lumen space where you just pumped a bunch of hydrogen ions in there through this little machine, turns the turnstile around, ADP and P gets put together, and the plant makes ATP. So just by using light to get electrons excited, plants can make their own ATP. So they don't start with a substrate like glucose. They just start with light. And that gets the ball rolling. Now, having said that, people often get confused about this, and they think, okay, plants are the things with the chloroplasts, animals are the thing with the mitochondria. So plants make their ATP with light, and animals make their ATP with glucose. So it is true, except that plants also have mitochondria. So when the lights go off, as we see right here, they're not making any ATP. And plants still need ATP in the dark. They don't just completely shut down in the dark. They need ATP to do all the things that any organism has to do. Pump ions, grow, defend against predators, all that sort of stuff. They do that with their mitochondria, and they use the substrate that they have made. We're going to see how they make their own glucose, and then they use that to break it down in their mitochondria, just like animals break things down in their mitochondria. So plants are the Mac Daddy. They can do everything. So they can make their own ATP when the sun shines, and then use that ATP to actually make some food. We're going to talk about that soon. And then when the sunlight is off, they use your mitochondria to use the food that they made to break down that food and also make ATP. So they don't depend on animals. Animals depend on plants. Okay, so don't be confused and think that plants don't have mitochondria. They absolutely do. Okay. So once again, the holes that leave photosystem 2 to start the ball rolling, they get filled by splitting water. This is part of why you have to water your plants. And this is all where the oxygen comes from in terms of the oxygen that early scientists noticed that plants were giving off. Okay, if we think about a pesticide, a common pesticide, and how it works, this common pesticide is paraquat. Paraquat is not legally used in the European Union anymore, but is still able to be used in the U.S. Before marijuana was legalized in a lot of states in the U.S., it was used as a pesticide on marijuana crops, and especially marijuana crops in South America, and that is supposed to be a pesticide that's specific to plants, but it turns out it works on humans too and can cause damage to humans. All right, so how does it work? What paraquat does is it steals electrons from cytochromes. So these electrons are falling down this chain. As they fall down this chain, it's used to move protons from the stroma into the lumen. Paraquat instead steals them away from this chain. So can't move the protons, can't make ATP. Okay? Plants have reduced ATP production. They don't get any NADPH produced as the terminal electron acceptor in photosynthesis. The PS1 holes that are filled from PS2 that are left empty, and that kills the plant. In animals, paraquat steals electrons from the cytochromes. So the cytochromes are the electron carriers in the electron transport chain stage 4, and they get passed down an energy gradient with oxygen as the terminal electron acceptor. It steals those electrons. So if you steal those electrons, you don't make ATP, and that will cause the unbound oxygen here to be reduced to an oxygen radical. This is oxygen with an unpaired electron. You've heard about free radicals doing damage to your cells. This free radical, because it's not accepting electrons from the electron transport chain, will grab electrons from whatever's near it and can kill off different parts of the cell by doing that. And that will cause cell damage. Paraquat tends to accumulate in the lungs of mammals, causes pretty bad damage. So this pesticide that was supposed to be specific to plants is actually affecting animals through a very similar mechanism because it targets the electron transport chain.